Well, welcome everyone to this new talk in this interesting series on Build a Cell. As you all know, a living cell is a highly regulated system, and building a synthetic cell then also, of course, entails building a control system, and more in particular, a vital element of all living systems, and hence also all living synthetic systems, is the capacity to coordinate replication with growth, and the replication of the DNA with growth and division. And that's precisely the task of the speaker of today, Mareike Berger. Mareike has been a student in my group, and she has been developing a model for growth and division and replication, which will be tested, of course, together with experimentalists. At least that's the plan. And today she will talk about the control module that she has developed, and how this is also inspired by the system that we all know so very well and that we take as inspiration, the bacterial cell E. coli. So Mareike, the floor is yours.
Thank you very much, Pieter Rein. I will share my screen. And you should be able to see my slides now. Is that correct?
Yes.
Wonderful. So first of all, thank you very much for giving me the opportunity to speak at this seminar. And well, as Pieter Rein said, I will talk about the cell cycle regulation in the bacterium E. coli. So what you can see here is a group of growing and dividing bacteria cells. And if you look closely and you follow one of them, then you will see a bright white spot light up at some point. And this is actually the moment when replication starts. And then replication finishes and the cell divides and a new round starts. And this ability to make autonomous copies of oneself is really one of the key features of every living organism. But up until now, it still remains poorly understood what molecular mechanisms give rise to such behavior. So how can cells make autonomous copies of themselves? Well, there are two ways of studying, of kind of investigating this question. We could either start with the living system and look at how it works and try to kind of manipulate it and understand it. But there is also a new field emerging, and I think it's summarized quite well by this quote of 𝗕𝗏𝗵𝗶 that says, what I cannot create, I do not understand. So the idea is to try to start actually from the building blocks and reassemble those building blocks and try to recreate molecular functions. And the hope is that with this approach, we will also gain some new understanding about biological systems, and especially about maybe what minimal features are required to get some biological functions. And this is exactly what we want to do in the basic project. So building a synthetic cell. We have already had one speaker in this seminar series from the lab of Kees Dekker. And it is actually a collaboration of different groups in the Netherlands. And the goal is a very ambitious one, namely to build a synthetic cell from the bottom up. And here you can see an artist's view of such a cell, where we have the three core processes, namely growth, DNA replication, and cell division. And so these processes need to be coupled, of course. So the idea is to start with the container and the genome and to combine those. And then the cell should grow by reading out this genetic code. It should be able to replicate the DNA and to segregate these two chromosomes. And then the cell should divide. And these three processes are coupled via the cell cycle. Now, what would happen if you don't have a cell cycle? Well, it could look something like this. We could start with some well-defined cell, but then if we don't control division, if we don't control replication, we could end up with very small cells which don't even contain a chromosome, or with cells that contain way too many chromosomes. So a robust replication cycle is crucial for the survival of every organism. But what are really the minimal requirements that the cell cycle needs to have? So one of them is homeostasis. So the cell cycle needs to make sure that these cells remain within a certain range of parameters, and especially with two parameters, or with two variables, namely the cell size. So the cell size needs to be maintained within a certain range of sizes. So for example, if a cell is born a bit larger than average, then if it would just double its size, and it would in the next cell cycle again be larger, and if there is again noise, it would just fluctuate and would not have a controlled volume. So there must be a mechanism that drives the system back to some size, some average size. And the strategy how to do this, one strategy how to do this is called the sizes strategy. So according to the strategy, the cell, no matter how large it is born, will grow until it reaches some fixed size and then divide. And of course, this will maintain a stable size, because it will always grow until this fixed size and divide. And then there is maybe some error, but it will again grow to a fixed size and divide. One other homeostatic mechanism that we need is the homeostasis of the DNA density. So what you can see here is what happens if the replication rate is 6 times as high as the doubling rate of the cell. Then the DNA density actually increases exponentially. And because with every doubling of the DNA, or with every replication round, we get 2 more DNA strands. So we're doubling with every replication round, and this is an exponentially increasing function, which would completely destroy such a synthetic cell. So we need to find a mechanism that ensures a constant DNA density. Then we also need to couple, of course, DNA and replication, DNA replication and cell division. So what we need to ensure, what the cell cycle needs to ensure, is that first the DNA is replicated and only then the cell divides. And so the question is, how do living organisms do these tasks? And we can have a look at different organisms. For example, we could look at the eukaryotic cell cycle. And the eukaryotic cell cycle is actually very complicated, even though it is highly organized. So we have certain phases that are separated via checkpoints, and these checkpoints are regulated by a very complex biochemical network. And to reproduce a eukaryotic cell cycle, we would need many, many genes and very complex interactions. So what we would rather like to do for a synthetic cell is to try to implement the bacterial cell cycle. And the bacterial cell cycle is simpler. It consists only of 3 phases. And so these phases are, the first is the growth period, so from birth until replication initiation. Then we have the C period, from replication initiation to the end of replication. And then we have the D period, which is the time from the end of replication until division. And these phases are rather loosely coupled, and they're not as strictly separated as in the eukaryotic cell cycle. So let's look at the cell cycle of E. coli. So in E. coli, it has actually this remarkable property that it can grow very slowly. It can take, for example, in poor environment, it can take 2 hours to double its volume, or in better growth conditions, it can grow up until 20 minutes, double every 20 minutes. And so in order to achieve such a variability, the time of these periods must vary. But experiments have shown that this C plus D period is actually rather constant. And this led to the idea that replication is really the moment in the cell cycle where control is happening. So the idea is that cells grow until they reach some critical volume, and then they initiate replication. And after that, the cell simply divides after a fixed time later. And so we can now have a look at how such a mechanism ensures stable cell cycles. So this idea was generated by 𝘊𝗺𝗺𝗍 because he combined in the 1960s this idea of exponential, or this finding of an exponential growth of the cell with the fact that this C+D period is constant. And he concluded that what the cell needs to do is to initiate at a constant volume per origin. So let's have a look at such a cell cycle. So what we can see here is the volume of a cell that increases exponentially in time. And at birth, there is a single chromosome with this yellow dot here, which is the origin region. And at this origin, replication can be initiated. Now, at birth, there is only one chromosome. So there is one origin. So the number of origins remains constant. And now the origin density is given by the number of origins divided by the volume and is plotted here. And the idea now is that replication is initiated when a critical origin density is reached. And when this critical origin density is reached, replication is initiated and the number of origins goes from 1 to 2. And this means that also the origin density increases by a factor of 2 and is again very far away from this critical density. And then the division can be coupled to replication initiation via this constant C+D period. And then we see that such a mechanism ensures that the cell always initiates at the critical initiation volume, and at division, both the volume and the number of origins are divided by 2, so that the origin density is actually not affected, and we obtain stable cell cycles. And but now the question is, does this also work at high growth rates? Because experimentally it's known that these cells can divide every 20 minutes, but the C+D period actually takes 40 minutes. So how does this go? Well, let's have a look. So what happens now is that these cells are growing faster. So that means that they reach this critical density faster. And then again, they initiate replication. The origin density is doubled, and the cell continues to grow. And contrary to what happened in the low growth regime, the cell can now reach this critical density within the same cell cycle. And now we obtain cell cycles that have overlapping replication cycles. So this is what you can see here. So the cell has already started initiation here, but then in the same cell cycle, it initiates again. And we have now 4 origins. And then the cell divides and we have again 2 origins. And then at the next round, it will initiate again. And we get a system where only by initiating at a constant origin density, the cell maintains again stable cell cycles. And so this idea to initiate at a constant density and to divide a constant time later ensures stable cell cycles over the full range of growth rates. So this would be a great way of implementing such a mechanism in a synthetic cell. Now, we have also more recent experimental results, because all of this was found in the 60s. And this thanks to some new single cell tracking mechanisms. So we can use now these experimental results from the mother machine, where single 𝗀𝗃 cells grow and divide, and we can track them at every time step and obtain these time traces now. And we can here see nicely that indeed these bacteria cells grow exponentially. And we can now look at the fluctuations from one cell cycle to the next. So for example, we can plot the volume that has been added from birth to division. And by doing this for many, many cell cycles, we can obtain a correlation plot. So this is what you can see here. We can plot this volume, this added volume, as a function of the birth volume. And now something surprising has been found, namely that the volume that is added is actually independent of the birth volume. And this is not what we would have expected, because originally we thought that these cells are maybe maintaining a stable size by always growing until they reach a fixed size. And this would be the sizer strategy. And instead, what cells do is to always add some independent volume from one division event to the next. But now, can this mechanism also ensure a stable cell size? Well, yes. So if you start with an initial perturbation that is very large, and then you follow these cells on average over many generations, then you see that this error is reduced by a factor of 1/2 with every generation. And so also the strategy of always adding some independent volume generates stable system. But how is this now coupled to the replication cycle? Well, we can also visualize the replication cycle. And indeed, what we see here is that we are in this overlapping regime that I mentioned earlier. So we have two origins at the beginning of the replication cycle. And then the time to replicate here is the C period plus the D period. And now we can visualize again or measure again this added volume from one initiation event to the next. And again, we do this for many, many cells and obtain these correlation plots. And like for the division volume, we find that cells seem to try to add an independent volume from one initiation event to the next. And so again, this strategy gives a stable system, because as we can see here, when we start with a large deviation in the initiation volume, always adding some independent volume will reduce your initial perturbation and bring you back to the average. So all of these observations are, of course, nice because they can guide us in finding a molecular mechanism, but they still don't really tell us what is happening on a molecular model, on a molecular level. So to summarize, I will now just summarize these experimental observations. And I actually didn't mention that one important observation, namely that the initiation volume is one of the most tightly controlled parameters if you look at different cell cycle parameters. So it varies actually only by 10%, whereas the growth rate and, for example, the time from initiation to division varies by 15%. So to summarize, we have found that the DNA replication is initiated at an on average constant volume per origin, and the added initiation volume is independent of the initiation volume. And finally, the DNA replication initiation is very tightly controlled. But so what does all of this mean? So we have now found all these experimental observations, but do we now understand how replication is initiated really on a molecular level? So this is the question that we now want to investigate. And for this, we need some modeling, because there have been many experiments, and the biochemical network of E. coli is very well known and it is very complex. But there is still lacking some model that can really explain how all of this is working together. But so far, there have been proposed two types of models that are qualitatively very different. So the first one is the initiator titration model. So the idea of this model is that there is an initiator protein, and you need to accumulate a fixed amount of this initiator protein in order to be able to initiate. And then from one initiation event to the next, you always need to accumulate a fixed number of these initiator proteins. And this is, of course, very similar to this adder idea. So this idea that we need to add an independent volume from one initiation event to the next. It's actually a molecular implementation of this phenomenological observation. And there is a very different type of idea, namely that there is a switch between an active and an inactive form of the initiator protein. So we will now go into both of these models and test whether they are consistent with these experimental observations. Let's first look at this initiator titration model. So the idea of this model is that there is an initiator protein, and the initiator protein can be bound to some titration sites on the chromosome. And these titration sites have a very high affinity. So whenever an initiator protein is produced and there are still some free sites, then the initiator protein will bind to these sites and therefore not be able to initiate replication at the origin. But when all of these sites will have been filled up, then the initiator can bind to the origin and a new round of replication can start. And so this idea we have modeled in a mathematical model where we assume that the volume grows exponentially in time and that the initiators are produced proportional to the volume. So the number of initiators increases, but as long as the number of initiators is smaller than the number of titration sites, replication cannot be initiated because all of these titration sites are bound. But when the number of the initiators is equal to the number of titration sites, this free concentration can rise and a new round of replication can be initiated. And as replication is initiated, these sites are being replicated, new sites are being produced, and this is what you can see here. Now, because these sites are distributed homogeneously on the chromosome, we see that the number rises linearly in time. And because the number rises faster than the number of initiator proteins, the free concentration drops and a new round of replication can be prevented. So we see that at low growth rates, this mechanism is actually a great mechanism to ensure stable cell cycle. And because it is already adding some fixed amount of proteins per replication cycle, because this fixed amount is set by the number of titration sites, this is already kind of reproducing these adder correlations. But now we need to look at higher growth rates. Because at high growth rates, we have already mentioned that the time to replicate the chromosome takes 40 minutes. But the doubling time is 20 minutes. So this means that the total number of initiators increases actually faster than the total number of titration sites. And in such a system, we obtain unstable cell cycles. So this is what you can see here. Again, like in the low growth rate regime, we grow until we initiate when the critical initiation volume is reached. But because now the number of titration sites is not fast enough being produced, we actually continue to increase the free concentration. And we have here implemented some fixed period during which the cell cannot reinitiate, which is called the blocked period, just to ensure that the system can kind of relax. But instead of reducing this free concentration, actually continues to rise, and we obtain severe reinitiation events. So titration cannot ensure robust cell cycles at high growth rates. And we can visualize this even more if we look at the coefficient of variation, because the coefficient of variation is 0 at low growth rates. But then as we reach this overlapping regime, this titration model completely has a very large coefficient of variation because these cell cycles are actually not stable and we continuously reinitiate. So the coefficient of variation exceeds the experimentally measured coefficient of variation, and this titration model is not sufficient to ensure stable cell cycles at high growth rates. So we need to look into this activation switch, so this other model that I mentioned. But how can this activation switch now reproduce this experimentally observed observation? Well, the idea is that there is a protein that can switch between an active form and an inactive form. And when the protein is in the active form, it can initiate replication at the origin. And now this switch should reproduce these experimental observations where replication is initiated when a critical origin density is reached. And how could this switch now do this? Well, we can have a look at the deactivator and the deactivators in the cell. And actually, we find that there is a deactivator that is located at the origin and is therefore, its concentration is therefore proportional to the origin density. And there is also an activator that is present at a constant concentration, which is namely these lipids, these acidic phospholipids. And if we now combine a deactivator that depends on the origin density with an activator that has a constant concentration, we can obtain an active fraction of this activator protein that is proportional, that senses the origin density. So this is what you can see here. So as the origin density decreases, this deactivator becomes weaker and the protein will be more pushed towards the active state. And therefore, this active fraction increases as the origin density decreases. And when replication is initiated at a critical fraction, then the origin is being doubled, and therefore the deactivator becomes much stronger, and most of the proteins will very rapidly be in this deactivated state. So we have now created a switch that really senses this origin density and is really proportional to the origin density. And so we can, with this switch, explain how replication is initiated at a constant volume per origin. So this is great. We have now built this activation switch, but we should think again, because what we have built here is actually a size sensor. And I have shown you in the beginning of my talk that we have observed these adder correlations. So there seems to be a mechanism that adds some constant volume from birth to division. And what would happen in this model here is that if you are, for example, born a bit smaller, then you would grow until you reach this critical initiation volume, and then a fixed time later you divide. And this is clearly not the adder, because you grow until you reach a fixed size, and this is not independent of your birth volume. So the question that we need to ask is whether our size sensor can now give rise to these experimentally observed adder correlations. And actually in our model, we can, if the activation and deactivation rates are really high, we can find a mapping between the active fraction and the volume of the cell. And this is really this idea of the switch, that when the volume is small, then the active fraction is low. And when we reach a critical volume, the fraction shoots up, and at a critical initiation volume, we then initiate replication. But this curve here depends on the concentration of the components of the switch. So if we, for example, have a higher lipid concentration, then this entire curve is shifted to a lower value. And if we have a lower lipid concentration, then this whole curve is shifted to the right. And this means that the initiation volume will also be shifted. So from this plot, we can obtain this inverse dependence of the initiation volume on the lipid concentration. And now what this means is, if we have fluctuations in the lipid concentration, we will directly also get fluctuations in the initiation volume, because the initiation volume is dependent on the concentrations of the switch components. And so what we need to think about is how do the fluctuations in the lipid concentration now have to look like in order to reproduce these adder correlations in the initiation volume that we have shown in the experiments. And so we decided to model the change in the lipid concentration in the most simple form that we can think of, namely by a constant production term. then a dilution term, which is proportional to the growth of the cell. So this means that the lipids are not being degraded. They are simply being diluted. So we produce lipids at a constant rate. And the only way how the lipid concentration can be reduced is by increasing the volume and diluting basically these lipids. And then the last term is the noise term, where we have a Gaussian white noise. And so using this type of differential equation, we can now see how a lipid concentration that starts with a high deviation relaxes in time. And in gray, you can see all these time traces of the different realizations, and they are stochastic. But on average, we see that they follow this differential equation with exactly a decay time of 1/2. So with every generation of the cell, the lipid concentration, so the error in the lipid concentration, is reduced by a factor of 1/2. And this is exactly the temporal correlations that the adder has. And so because we have now a mapping between the lipid concentration and the initiation volume, which is directly inversely proportional, the initiation volume will have the same temporal correlations as the lipid concentration. And this now can give rise to this adder correlation that has been observed experimentally. So even though we have built a size sensor, we can nevertheless explain these experimentally observed adder correlations. So now we have an open question. Namely, I have shown you that there is this initiator titration model, which works at low growth rates, but not at high growth rates. And I have also shown you that there is this protein activation, which can also explain these adder correlations. But so the question is, why are there now two homeostatic mechanisms? And the second question is, of course, how are they coupled to each other? How are they, which of the two is the main mechanism, or how could they help each other? And so for this, we have extended our switch model, and we have included all known activators and deactivators. And I will not go into the details now, but I will show you how the time traces look like of such a system. And what we see is that this active fraction is not so nicely behaved. It's actually decreasing very strongly, but then it's going up very rapidly, and the slope that it has here is rather low. So this is not a very good sign if we want to have a robust system. If we want it to be robust, it should increase rather sharply here when it reaches the critical fraction. And at high growth rates, the system actually produces very nice oscillations. And so if we look now at the variation in the initiation volume, we see that as a function of the growth rate, at low growth rate, the switch is actually above the experimentally observed variance. So the switch is not very robust, is robust at high growth rates, but not at low growth rates. So the switch would actually need an additional stabilizing mechanism at low growth rates. And here we can now say, hey, wait a minute, we have the titration mechanism that works pretty well at low growth rates. And we have now the switch that works well at high growth rates. So why don't we combine these two mechanisms? And this is what we actually did. So we combine the activation switch now with titration sites, and we now initiate replication at a critical free concentration. So we combine these oscillations between bound and free with the oscillations between active and inactive. And so the idea is that titration will increase the sharpness of the switch at low growth rates, and the activation switch will prevent these reinitiation events at high growth rates. And this is exactly what you can see here. So as a reminder, the titration mechanism is this green line, and that shoots up at high growth rates. Then the switch model has a high coefficient of variation at low growth rates, but then a low one at high growth rates. And if we combine those two, we see that at all growth rates, this really leads to a very low coefficient of variation, which is very comparable to the experimental results. So the full switch titration model ensures robust replication initiation cycles at all growth rates. And so with this, I want to summarize our results on the replication regulation in E. coli. So we have found two homeostatic mechanisms that regulate replication initiation in E. coli. First, there is this initiator titration idea. And second, there is the switch idea. So the initiator titration, again, is this idea that proteins can bind to the chromosome or they can be free, whereas the protein switch has this idea that a protein can be in an active or an inactive form. And we have shown as well that this protein activation switch, even though it is really sensing the size of the cell, can also give rise to these adder correlations by introducing noise in its components. And then we have found that the combination of these two mechanisms can actually give rise to robust cell cycles at all growth rates. But now we want to return to our original question. How should replication initiation be regulated in a synthetic cell? And for this, we suggest actually that the titration mechanism is the best, because it is a rather simple system where we only need to produce an initiator protein. And what we actually are doing at the moment is to collaborate with the group of 𝘂𝗏𝗏𝗏𝗏 and together with Lorenzo, who is now trying to build a E. coli cell where we replace completely the replication regulation that is native to E. coli by a system that we construct. And this is supposed to be a testing station for then moving on to our synthetic cell. And the idea that we have is to have an initiator protein that can be regulated by some regulator, so that we can also vary this initiator protein concentration. And then we want to modify also these titration sites and test different numbers. We want to change also the location and bring them more to the origin. And finally, we will also then have this initiator protein be able to initiate replication at the origin. And so the idea is to then have this module, which we could later implement also in the synthetic cell. And so with this, I just want to mention some open questions, because of course, I now made it sound like everything's clear, but I've skipped over a few things. So for example, one really big question that remains to be solved is, how is division regulated? And especially, how is it coupled to DNA replication? So I have always assumed that there is a timer from replication to division, but the question is, of course, how do you implement that molecularly? And one idea would be to have some division protein that is located at the terminus, and that the doubling of this gene at the terminus we could amplify this and then send a signal for division that is really triggered by the end of replication. So this would be a way of implementing a timer. Another idea would be to use the fact that we also need to segregate the DNA and to couple then a DNA segregation to cell division. But there is also currently a lot of debate in the community of E. coli how much this timer idea is really valid. And there is another suggestion, namely that there is an independent adder from birth to division via the protein FTSZ. So this could also be a possibility to not use this timer, but then really have replication and division kind of separately regulated. And yet another idea is that you could implement an adder from the moment of replication initiation to division, which is also currently debated in the on the question of how E. coli regulates division. Then one important question is, how can immediate reinitiation events be prevented? And I didn't mention this, but there is a protein called SEC-A, which can block reinitiation at an origin that has already initiated. And so the question would also be whether in a synthetic cell we need such a protein or whether it is not absolutely necessary. And another question is, how should these titration sites be localized on the chromosome? Would it be better to have all of them at the origin, or is actually this homogeneous distribution maybe a better way, which is the one that 𝗀𝗏𝗺 chose, but we don't really know why they are located all over the chromosome and what the advantage would be of that. And finally, also one really interesting question is, what will be the minimal system that is required to ensure stable cell cycles? So would titration really already be sufficient, or do we need this additional switch? Also the question of which one of these different couplings to division is the most stable one? And I think all of these questions are still really very much open and are still many interesting questions that we will be thinking about in the future. So with this, I want to thank you very much for your attention. I want to thank Pieter Rein and Wolde and all my colleagues, and I'm open for questions.
Thank you, Mareike. Thanks for the very nice talk. So the floor is now open for discussion. Already a number of questions have been raised in the chat, so I'll have a look at that. So 𝘕𝗏𝗵𝗆, could you pose your questions? You have several, so please go ahead.
Okay, thank you so much. This is an amazing talk. I learned a lot today. So the first question is a very simple question. Your first video, which one was labeled for imaging? So I guess, original replication, that image, or the labeled or something else, a label. Yeah, that one.
Yeah, so here I mentioned what it is. So it's DNA-Q, which is a subunit of the DNA polymerase. And
I missed that part.
The standard way of, at the moment in the field of labeling replication, so that you can really see when replication is active, because it is basically this subunit of the DNA polymerase that is being labeled.
Okay, so I'm sorry that I missed that part. So I just fascinated by your movie, so I didn't look at
that. Yeah, I didn't mention it. I just put it, so no worries.
Okay, and then my second question is, so one thing I don't understand mechanistically is that origin density sensor basically detect 1 to 4, I believe, maximum, depending on how fast 𝗀𝗏𝗺 divide the per volume or per cell, and then which probably is so stochastic, because we're talking about one molecule or one location to up to 4, would be very, very low number. So that kind of causing a lot of stochasticity. And then, however, cell control is very, very precise. In terms of this one. So I kind of fascinated by the how cell control, but I still don't understand how that is achieved.
Yeah, that's a very interesting question. I really like this question. So you can, of course, go into these two mechanisms and then wonder which one of those would maybe be better in indeed doing that, because you want to sense, like you say it, some very small number, right? You say the origin is going from 1 to 2 or from 2 to 4, and can actually also go from 4 to 8 and 8 to 16. So it's really a very precise sensing. And well, in this slide, you can, of course, see the origin density, that it really has this sharp change here. And the question is how sharp can, for example, the switch now map this? And so here, what we did is we choose very high rates of activation and deactivation. And then this follows it really very, very sharply. And I would say this is a good sensor for this very low change in the numbers. because we have this very active system that is driving it via activation and deactivation. And the number of this DNA A is in the order of 1,000. So this would be a better, like a larger number with less fluctuations. So the point is, however, if you reduce these rates, this also becomes weaker and kind of flatter. So then the sensor would become less precise, I would argue. So it would be very interesting to kind of test these ideas. I think it's still very hard to measure really this active fraction experimentally, which is why all of these modeling still are, yeah, kind of possible, and yeah, await experimental verification. But maybe just quickly to finish on the question here, what you can see is here, this would be the other type of sensor where we have the free concentration, which also rises relatively sharply and reduces very sharply as well. So this is in the titration model. And the idea is that we amplify the very low number of origins by a large number of titration sites that will then be coupled to the number, because as soon as you start replicating, it will kind of, many processes will follow from it, like, for example, replicating all of these new sites. And this would also be a way of amplifying kind of this very small number of the origin that changes.
Yeah, so, in general, I understand, because I mean, this is sort of the sequestration type of mechanism. In that way, kind of once you kind of pass the threshold, you will have the huge jump, two different states. So that kind of makes sense. But what I don't understand is we are not talking about one molecule or two, and then that and also many things in the system. We are not talking about many of the components in the system. Also have lots of noise. I mean, in terms of number of the molecule in the system. So if we kind of combine every kind of thing, all the component, let's say plus minus one molecule, whatever, and that kind of lead to, I mean, huge kind of noisy system and the stochastic kind of system, but still cell kind of control our division. And the E. coli still control the division. So that part I still don't understand. By even sequestration kind of mechanism, titration mechanism, even though that kind of clear kind of jump, I mean, I understand that part, but the other part, how one molecule and two, how they kind of differentiate between this kind of very small number kind of change.
Yeah. So I think one additional mechanism, is, of course, this blocked period via the protein sec-A, which really blocks the origin for 10 minutes approximately. So this gives you also some kind of refractory period to kind of start all these other processes. So this might be a very, very important mechanism, which would be also super interesting to test in a synthetic environment to see how important this is. Because in E. coli, if you remove this sec-A, it almost always leads to reinitiation events. So it really might be that this is one of the most crucial points about achieving this high sensitivity, that you just need some time that the system can switch from this high potential of initiation to a really low potential, so that you kind of amplify this sensing in a way.
Okay, actually I have one more question, but I'll keep other people kind of asking question, and then I'll come back to my last question later on. So probably that's better for other people. Thank you so much. It's so fascinating. It's amazing.
Thank you.
Thanks. Okay, so 𝗰𝗆 Bordeaux, could you spell out your question?
A lot of the time people don't unmute themselves. I don't know why, but you might be forced to read the questions for them.
I'm happy to read the question of 𝗰𝗆, but maybe 𝗰𝗆 wants to phrase the question herself, maybe? I'm happy to read it. Okay, so the question is, what is the initiator protein? Is it a protein or a small molecule?
Yes. Okay, so I don't have a picture of it actually, but it is a relatively small protein that consists of different units. And for example, it has different domains. One would be the binding domain to the DNA, which has been identified to have this high affinity to the chromosome. Then there is another domain that allows ATP and ADP to bind, which allows it to then switch between these two different states. Yeah, that would be my answer to your questions. I don't know.
Okay, Emma, are you satisfied? Okay, I'll go to the next question in the list. So Alexander Nover, would you like to raise your question? I mean, I can again read out what was typed in the chat, but I think it's nicer if the participants raise their question. Okay, well, let me then read the question out aloud. Does similar initiator model apply to crazy fast dividing cells? So does this model apply to both slow and fast growing cells? I'm not entirely sure what Alexander means by crazy fast dividing cells, but
yeah, so I guess I think the fastest observed growth rate in E. coli is maybe like 18 minutes, which would maybe be crazy fast. Indeed, it is very fast. So I would say this is exactly the kind of question we try to answer with our model. And we find, for example, that the initiator titration model is clearly not able to ensure stable cell cycles at this crazy high growth rate, whereas the activation switch is exactly almost built for the fast growth regime, because there are, like you can see here, different processes or different activators and deactivators, and they are actually located at very specific locations. And in our model, we show that these locations really help, especially in the fast growth regime, so that there are very specific timings. So for example, you have the very strong deactivator RIDA that is, especially at high growth rates, will bring down this active fraction very quickly because it's a very potent deactivator. But then at high growth rates, if you want to already reinitiate after 20 minutes, then this deactivator is still active. So you need a strong activator. And for example, this DARS2, it has been shown that it even has a growth rate dependence, and that is especially active at high growth rates. So we show in our model that also the position of this site matters and really helps in raising this active fraction again quickly in order to be able to reinitiate at such a high rate. So I hope this answers the question.
I think it was clear to me. Yeah. If not, raise your voice. No? Okay, then I continue. Brendan Mortesh, would you like to ask your question? Okay. Okay, so the question is, would you speculate how general this model is? Would it apply to all bacteria or prokaryotes, or can you predict some exceptions already?
So we have compared it to other bacteria a little bit. So actually the feature of having these overlapping replication forks and being able to grow so quickly is very specific to E. coli. So this, I would say, does not apply to E. coli, but especially this titration mechanism, for example, has also been shown in plant cells. So even in eukaryotes, this titration mechanism seems to play a role. And DNA is also, the protein DNA-A, which is the activator in E. coli, is also a well-conserved protein in other bacteria. So I think Bacillus subtilis and maybe even Caulobacter, I'm not 100% sure, also have DNA-A, but not necessarily with the activation switch. So different bacteria maybe use similar classes, of regulators, so either maybe titration or protein activation, but I would not say that we can directly map our model now to any other bacteria.
Okay, thanks. Thanks, Emma Jones. Would you like to raise the question yourself? I think it's nicer. Okay, I'll read it out. For your replication in synthetic cell process, why do you use 𝗀𝗃𝗃 as a platform instead of simpler cell like mycoplasma? Is it easy to use and presence of genomic tools, or for some other reason?
Yeah, I think we did start with 𝗀𝗃𝗃 just because it is extremely well studied. So there is, like I showed in my slides, the first experiments date back to the 60s, even 50s. So there is really a long history of trying to understand how replication initiation works. And as 𝗀𝗃𝗃 is really one of the model organisms, and it is still not understood, which is surprising because really there are decades of studies and there is still no model that can really explain all experiments and is consistent. We found this extremely interesting, so we really wanted to understand how 𝗀𝗃𝗃 is doing it. And then also because our collaborators, so I mentioned also the group of 𝘂𝗵𝘆𝗖, they have a big expertise on 𝗀𝗃𝗃, and you can very nicely, for example, introduce new genes, modify these titration sites, include a different initiator, and keep this bacterium alive. So yeah, that was our main motivation for using 𝗀𝗃𝗃.
Okay, thanks. Next question, Pieter Bandel. Would you like to raise your question? If so, please unmute. Okay, well, so the question is, would it be possible to hack this system and use it to over-amplify plasmids in 𝗀𝗃𝗃, effectively turn even low copy number plasmids into high copy, and amplify plasmids for cloning much faster?
Well, okay, so I don't really know how to answer this question. I think well, plasmids, they have their own control systems, so they are called plasmid control systems, and they also need to ensure, like I mentioned in the beginning, you need to ensure the homeostasis. And so also plasmids have systems to maintain a stable DNA density. And they also use similar mechanisms, either titration, or they use something where they block the next round of replication by binding of two different origin regions. And I think you can also manipulate these systems by playing around with the parameters. So I wouldn't know why you want to use the 𝗀𝗃𝗃 one for plasmids. Yeah, I don't know. You have to kind of specify your question. I'm sorry, so that I can give a more precise answer. Or let me know whether you already answered it or not.
Well, do you want to respond to this, Pieter Bandel? Okay, if not, let's continue then with the next question by Patrick. Patrick, would you like to raise your question? If so, please unmute. Okay, it's actually an interesting question. So what happens if you stop DNA replication? Would it stop cell division too?
Yes, very interesting question. So there are experiments. So you can postpone, for example, the initiation moment. So you could, by changing activators and deactivators, you can, or the DNA concentration, you can vary this initiation volume. And it has been shown that its relative division seems to be independent at this very small changes. So which is why in the end, I mentioned that maybe there is actually rather this double adder, so that you have one control on replication and one on division. So they seem to be less coupled than originally believed. But if you completely block it, then the system will even stop growth and have an SOS system that prevents to continue to divide. So I think like small perturbations are possible, but as soon as you would really completely block it, I mean, there are also even other mechanisms how you could recover from this, but in principle, cells cannot continue to grow and divide without a chromosome or without replication, as far as I know.
Okay, thanks. A question by Alexander Nover again. Alexander, would you now like to raise the question? Okay, so could the activation switch hacking help overcome cell cycle timer limitations if you activate DNA replication in E. coli very slowly? Could you make cells divide very slowly too? And conversely, could you speed up cell division artificially by speeding up DNA replication?
Okay, so this is about the question between the link of kind of growth and replication. And so yeah, I didn't insist so much on this, but I think what typically the growth itself is rather independent of the replication. So you could replicate at a higher rate and the growth rate is not necessarily affected unless you really completely kind of destroy your system and you have way too much DNA, then there will be a system kind of coupling it back to the growth of the cell. But I think the main direction how things are coupled is that you have growth, and that really depends on the growth environment. So for example, on how many nutrients there are in the environment, and that will determine how many ribosomes you have and how fast you grow. And to this, then replication couples via sensing, for example, the volume or the origin density, and division kind of couples, and that we're not really sure of, either by coupling to replication or also independently to growth. But the coupling from replication, so the growth is rather independent of replication and division cycle, unless there is some really severe perturbation.
Okay, thanks. Harold Murel. Would you like to pose your question? I think it's nice if people really unmute and join the discussion and provide feedback also to the answer, challenge the speaker. Okay, so let me read it out loud then. Could this possibly be an antibiotic target inhibiting DNA replication by sequestering or inactivating the initiator?
That's a very good question. This is not really my field of expertise, so I don't know, but it sounds like a very interesting idea. And it could be that there are already antibiotics that do that to inhibit replication. I'm not really sure.
Okay, I'm just checking the other responses in the chat, but I think the response to the question. Yeah, here I have a question. Justin Kipnis.
Oh, I can ask it.
Good. Great.
I actually have two questions. My first question is, for the initiator proteins, if an E. coli produces those, can it be secreted and also like bind to initiator titration sites on neighboring E. coli, or does it only affect that individual E. coli?
So you mean if E. coli really secretes it to the outside, to other E. coli cells, or?
Yeah.
I have never heard about this, so I'm not aware of this possibility. I don't think it secretes it, as far as I know.
Okay. And then my other question was, and you mentioned something about there's some mechanisms in like plants for titration. Are they conserved mechanisms, or they're just like mechanisms that evolve separately that are similar?
You mean similar to the one in E. coli?
Yeah, but are they the same like proteins or different proteins?
Yeah, so I think eukaryotes and prokaryotes, they are so E. coli and this specific plant paper that I read, I don't think these are the same or conserved proteins, but I'm not sure about, so yeah, like I said, so in bacteria, then DNA A can be conserved, but then I think within eukaryotes, they might also be groups that are conserved. So I'm not very confident about talking about eukaryotes so much. I would say, yeah, prokaryotes and eukaryotes, I would say, are not conserved, and within prokaryotes, they definitely can be conserved, and within eukaryotes, I'm not sure. Does that answer your question?
Yeah, thank you so much.
Thanks for asking the question, and thanks also to everyone else. I really appreciate your questions.
Okay, well, we're not done yet.
So we have more time?
That's a good question. Do we still have time?
So I have two more questions. I guess I mean everyone just everyone's question is answered right now. So I want to ask two more questions if we have time.
Yeah, so if you can raise your question quickly, and then also maybe Mareike, please give a quick answer.
Okay.
Yep.
So the last two question is basically think about let me find those. Oh yeah, think about the the situation. For example, you introduce double strand break using Cas9 whatever, and then want to see homologous combination E. coli without adding DNA into the cell. That's kind of typically people do for the genome engineering. But let's say you have the two cells. One is the fast growing cell, and then the other is the slow growing cell. And then I just wondering which would have higher homologous recombination rate, given more genomes in fast growing cells. Of course, per cell, and then the other factors. Other molecule recombination factor and so on. This is probably not your expertise area, but I just curious. Based on the number of the chromosome per cell, and we have the more template for the DNA recombination in the fast growing cell versus the slow growing cell, and you might not have the answer for that question, but I'm curious because I'm using something. I have some system. I can cut the DNA completely, and then I kind of have some observation, but I wanted to have some expert opinion about my observation. So so my system, for example, if I cut the DNA, the chromosome, fast growing cell die very quickly. But if I kind of have the very, very slow growing cell system using very, very limited nutrient and so on, the cutting and then dying rate is kind of a little bit delayed in a sense, but still, complete killing at the end of the day. I kind of have some idea, but I kind of want to confirm by the expert like you.
I'm not sure whether I'm an expert on this. I would
In
guess.
the case, that's fine. In the case, that's fine. I'm going to go to the next question. That is more, kind of your, area. So let's say, I observe, also many people observe in the field, synthetic biology or metabolic engineering field, some burdensome condition like using overexpression and so on, often lead to cell size growth without division. So what would be the general mechanism for that? So, think about, you express some protein, and then in some condition, and some cell become very, very big, without division. And then that happened many different cases. I just want to understand what's the mechanism.
Yeah, okay. So I think I do know one experiment where they did that, where they express some kind of useless protein, overexpress it. And then they also see that these cells grow bigger. And this actually corresponds well to a model where you say that there is indeed an accumulated protein, and people tend to believe that that is 𝗕𝗦𝗆 in E. coli, that is just produced via, I don't know whether you're familiar with these proteome sector models, but if you overproduce another protein, then there is kind of less capacity to produce this protein, this division protein. So you kind of reduce the fraction of this protein sector, and then you produce it as a smaller rate. So you will need to grow larger to reach the threshold, the fixed threshold. So this could be one way of explaining why these cells grow bigger when you overexpress some other protein.
So you are saying, the resource competition basically limiting the division-related, protein number until whatever concentration reach, and then that basically lead to no division.
Yeah. So if it's a fixed number that you need to accumulate, and you have a smaller concentration, then you need to grow for a larger volume. So the concentration is the number divided by the volume. And if the concentration is smaller, you need to grow for a larger volume to attain the same number.
That makes sense. But another thing is, if that is the case, the same competition idea also applicable to genome replication machinery. Why that is not true for that one, but only true for the division protein?
Yeah, okay. So it could be that the cell is pretty optimized at this gene concentration. So because you need a certain number of genes to be able to express really very efficiently. So it could be that the cell size doesn't really matter so much, but that the gene concentration really matters for the cell. And that
no, my question is not that one. So you're talking about division related protein is expression limited by overexpression of useless kind of GFP protein, for example. But same argument will be true for replication related proteins. So that protein also not much produced.
I did understand your question correctly, but it could be that there are, for example, you could imagine that there is, if for example the if the protein is regulated, for example, negative autoregulation is a way of kind of sensing your own concentration. And if there is an effect that kind of tries to reduce your concentration, you can have active regulation to increase it again. So there could be a mechanism for example, a replication-related protein to nevertheless maintain a constant concentration. And that could be because it's maybe more important to be at this gene density, whereas it's maybe less important for a division protein. That would be it's just an idea.
Yeah, that makes sense. So the completely makes sense. So every single different gene has a different way to regulate, feedback, and so on. So that's fascinating. Thank you so much. It's amazing. I learned a lot. Thank you.
Thanks.
Okay. Well, thanks. Thanks, Mareike. Thanks to all the people that raised their question. I think we had a nice discussion afterwards. Well, thank you to all of you, and hope to see you again in the next entry in this series. Bye-bye.
Thanks a lot.
Thanks, everyone. Bye.
Bye.
Bye.